Hello students, welcome to Vihira Academy. I am your educator Dr. Hina Devnani and today I will be teaching you chapter number 5 of 12th standard biology. The name of the chapter is Principles of Inheritance and Variation.
Now, before starting the chapter, let us understand some basic terminology. First is genetics. Genetics is the branch of biology which deals with the transfer of characters from one generation to another generation. In genetics, we study about heredity, inheritance, variation, gene transfer, and many other things. Now, what is heredity? Heredity is the process of transfer of characters from one generation to another. In reproduction, what happens? The parents give birth to young ones which are similar to them. They are similar because of the transfer of characters. So the parents have this capability to transfer their characters through genes, or we can say through DNA, to the next generation. This process is called as inheritance and the phenomena is called as heredity. Now during inheritance, what happens? When the DNA or the genes are getting transferred from parents to offsprings, some minute changes occur in them. And these changes lead to behavioral and physical changes in the offsprings. And this leads to our last important term, that is variation. After understanding the terminology, let's focus on the basis of heredity. I've written a name over here, Johann Mendel. He is also known as the father of genetics. Why he is known as the father? Because in the year 1866, he gave a theory, he proposed a theory by doing a set of experiments for 7 years. He said that inheritance occurs due to certain factors. These factors are now called as genes. In those days, he was not knowing that these are the genes. He just mentioned certain factors are responsible for transfer of characters from parents to offsprings. And these factors or these genes, as we know now, are present in our chromosomes, which are present inside our cells. So this is the basis of heredity. Gregor Johann Mendel was an Austrian monk. He had studied hybridization in plants, and the plant that he chose was Pisum sativum, that is the pea plant. There are certain reasons for choosing pea plant. First reason was it was very easy to grow in fields. Mendel had a field and he used to grow pea plants in that field. Second reason, the lifespan was very less, so easily he can grow the plants, harvest it and get the results. Third, it was very easy to hybridize these plants. Self-fertilization as well as cross-fertilization, both the processes were quite easy to carry out. And the fourth and the most important reason of choosing pea plant was it has 7 pairs of contrasting characters, which made hybridization experiments very easy. So now let's have a look at these 7 pairs of contrasting characters. As you can see on the board, I've written all the 7 pairs. First is color of seed. There are 2 variations of color of seed, yellow and green. Shape of seed, that is round and wrinkled. Flower color, violet and white. Color of pod, green and yellow. Shape of pod, inflated and constricted. Position of flower, axial and terminal. And height of plant, tall and dwarf. So in all the 7 characters, there were contrasting pairs, and this made it very easy for Mendel to carry out hybridization experiments. One more thing here was, in each pair, one character was dominant over the other character. Now what is the meaning of dominant? Dominant means a character which suppresses the other character when they both are present together. So the one which suppresses is called as dominant and the one which is getting suppressed is called as recessive. So in all these pairs, one character, the character that I have written first, the characters in each pair were dominant, and here the second ones in each pair were recessive. For his experiments, Mendel had to carry out cross-fertilization among the pea plants. the flower of the pea plant is bisexual in nature. That means it has the male and the female reproductive part, which is the androecium and the gynoecium in the same flower. So Mendel had to carry out emasculation and bagging to avoid this self-fertilization. What is emasculation? Emasculation means removal of anthers from the flower in its bud stage. That means when the flower is closed, it is in its bud stage. At that time, the flower will be opened, and with the help of forceps, the anthers will be removed and the flower will be allowed to close again. When the flower matures, it will open and it will have only the female part in that. Now, as soon as the flower opens, the second thing is bagging, which needs to be done. When the flower opens, at that time, the stigma or the female part needs to be covered by a bag. This will avoid any other foreign pollen grain to fall on the stigma. So these two methods were carried out by Mendel. Now, when the flower is getting matured and when Mendel was ready to carry out the experiment, he removed the bag, he dusted the pollen grains, the required pollen grains on the stigma, and then covered it back until fertilization took place and the fruits were there. So by this method, he carried out hybridization. Now in the next section, we are going to study about hybridization techniques which are called as monohybrid and dihybrid crosses, which were carried out by Mendel. The first experiment done by Mendel was inheritance of one character, also called as monohybrid cross. Now we have already seen 7 pairs of characters. This experiment was done for each pair of character. We are going to study the character of stem height. As we have seen, stem height characters has two variations, tall and dwarf. So we are going to use that character in this cross. What Mendel did in monohybrid cross, he took a parent which was tall and another parent which was dwarf. He took female to be tall and male to be dwarf. What he did, he crossed them. Now here you can see for tall I have written capital T, capital T. First of all, why I have used capital T? To differentiate dominant from recessive. Dominant is generally expressed by capital letters and the same letter but small one is used for recessive one to avoid confusion. We can also use D over here, but that is going to create confusion. We will not understand that this D is used for which character. If same letter is used, we can understand that yes, this is used for stem height. So that is a general practice in genetics. You use capital letters for dominant and the same letter is used for recessive but small one. Now why I have used capital T, capital T? Because genes are always present in pairs. These are known as alleles that we are going to study in later sections. So we will write capital T for tall and small t for dwarf. These are the genes which are present in our somatic cells. Now what happens? During sexual reproduction, they form gametes. So let's say this gene has formed gametes and in the gametes only one pair of the pair of allele will enter. That means if there are two capital T, one capital T will come in one gamete and another capital T will come in another gamete. So we can say two eggs are produced. In one there is capital T, in another there is capital T. Now if we talk about these male gametes, same thing, two pollen grains will be formed. Here there will be small t and here also there will be small t. In sexual reproduction, the mating is random, that we all know. So what will happen? Let's assume that this capital T mates with this small t. So what do we get here in the zygote? One capital T, one small t. Second possibility is this capital T with this small t. Again we'll get one capital T and one small t. Third possibility, capital T, small t. And this will be the fourth possibility. So that will also be capital T, small t. Now as you can see that by random mating there are 4 possibilities of zygote formation and all the 4 possibilities consist of same genes, that is capital T, small t. Now as I told in the beginning that dominant character is that character which suppresses the other character when both are present together. So you can see here capital is for dominant and small is for recessive. So capital T, small t, both are present together. So which one is going to express? The capital T one. That means the tall one. So when Mendel studied this, when Mendel got the results, he was really shocked. Why? Because what he had imagined, that if I carry out hybridization between a tall plant and a dwarf plant, I will get plants which are of medium height. Obviously, even we will think of the same thing. If a tall and short is mated, a medium height individual will be formed. But what happened here? All the offsprings, that means all the plants in this generation were tall. And because of this reason, Mendel was amazed. He was shocked. So, this was the monohybrid cross in which we have concluded till first generation. Now, what are these called as? This generation is called as P generation or parent generation. And the first generation is called as F1 generation. You can also call it as first filial generation. After this, Mendel carried out another cross and he wanted to see what happens in the second generation of the offsprings. So what he did, he took parents from this generation. In this generation, all the parents were same, that is capital T, small t. So he took this as male, female and this as male. So again, like how we did in this, we'll do in this. Two gametes will be formed. One will consist of capital T and the another will consist of small t. Again here also, one will be capital T and one will be small t. So these are the gametes. Now there will be random mating. This capital T will join with this and it will give rise to capital T, capital T. This capital T will join with this and it will give rise to capital T, small t. Again, this small t will join with this and it will give rise to capital T, small t. One thing that you have to note here is we have to always write capital first and then small. You can see here first small is coming and then capital is coming. But what I had written, capital letter first and then small letter second. Next, last mating is small t and small t. So as you can see here, we have got different combinations. First one we have got as capital T, then we have got capital T, small t, again capital T, small t and then small t, small t. Now what happened, when Mendel was carrying out this cross, he thought that he is taking all tall plants. So again he is going to get all the tall plants. But what happened here, again he was shocked, he was amazed. Why? Because he got 3 plants which were tall and 1 plant which was dwarf. The parents were tall, but still he got a dwarf plant. So this was an amazing experiment. Now, how do we come to this conclusion? Let's see. You can see here capital T. As we have seen earlier, capital T was tall. So I have written tall over here. Capital T small t consists of one capital letter, one small letter. Capital letter is for dominant. So if dominant and recessive are present together, dominant one is going to express itself. So this will be tall. This will also be tall, capital T, small t. This is small t and small t that we have seen over here. Small t and small t is always dwarf. So what he got here? 3 tall and 1 dwarf. This generation is called as F2 generation. F2 means second filial generation. So this was the experiment done by Mendel, in which he concluded that in F2 generation you will get the ratio of 3:1 for dominant is to recessive. So today we are going to stop here, and based on this experiment, Mendel gave certain laws which are known as Mendel's laws of inheritance. These laws and the next experiments, that is dihybrid cross experiments, and also certain exceptions of Mendel's laws, we are going to see in the next video. Till that, thank you for listening and keep studying. Take care.